Hello everyone, welcome once again to another tutorial session in bioinformatics. In today's video, I am going to show you how you can annotate bacterial genomes using the tool PROKKA. Now, when we talk about genome annotation, this is the process of identifying, labeling, and assigning functions to features on a genome. So in this tutorial, we are going to use PROKKA to perform the genome annotation. And so what we will do is to first download and install PROKKA. After that, we annotate a fasta sequence using PROKKA. We will download the sequence and use it for this exercise. And then we are also going to explore the output files that are generated by PROKKA. Now, in order to use PROKKA, you need to make sure you have a Linux PC because PROKKA works in a Linux environment. If you are using Windows, I have a video that shows how you can set up a virtual Linux on your system, so please watch that. The link is in the description box. In addition, you need to have Anaconda installed on your Linux system. We will use Anaconda to install PROKKA. And I have a video that shows you how you can also install Anaconda in a Linux environment. The link is also in the description box. Lastly, you need to have your sequence. The sequence that you are going to perform the annotation on. For this tutorial, we are going to use a publicly available dataset, so you don't have to worry. There's a sample online that we will use. Now that you are done, let's begin the activity. So after installing Anaconda, the next thing you do is to add channels. We are going to add 2 channels for this tutorial. Let me show you how. We say conda config add channels and we indicate the channel name. We begin with the first one, which is Bioconda. Let's add a second channel, which is conda forge. Conda forge. Now we have the 2 channels added. Let's confirm that the channels have been added. So we say conda config show channels. So we have the channels added for us. The next thing to do is to create an environment and install PROKKA in it. This is how we do it. So we say conda create we add -n for the environment name. So let's say the environment is called PROKKA. Let's use this. Then you specify the tool you are going to install. And then you also specify the channel. So let's say -c, the channel, Bioconda, and then -c, conda forge. And then we indicate the tool itself, which is PROKKA. So we've added all of them. So this will create the environment, which is called PROKKA. And then we're also going to install the PROKKA tool. And then we are going to install the tool. And we want to download, we want conda to check these channels and then download the PROKKA tool from it. So that is what we've just done here. So once you have this done, you run the command. And then you wait for the installation to proceed. You'll be asked to confirm. So you wait and then you confirm and install. So we confirm. Please note that sometimes from the entry of the commands, from the start of the entry of the commands for the installation, it will take some time before you get to this page where you confirm. I think mine took about 10 minutes or so. So just be patient and then wait for it to resolve. So we type Y for yes, and then we proceed. Conda also installs automatically all dependencies. So that is why I recommend you use it to install PROKKA. So let's install PROKKA now. So we wait for the downloading of the packages and their installation. So now we have PROKKA successfully installed. So let me clear the screen first. So the next thing we do after we have installed PROKKA is to activate the environment, and we activate it in order to use it. So this is how we activate a conda environment. So we say conda activates, and we indicate the environment's name, in this case PROKKA. So now the environment has been activated, and this is how it appears when you activate it. So we have the name of the environment also appearing on the prompt. So we have it there. Now let's test PROKKA. Let's issue the command PROKKA. Once you issue it, let me redo it again. PROKKA. Then we run it. Then we are going to have this list of statements appearing. These are the help statements that tells you how to use PROKKA. There are a lot of options here, but we are going to use just a few of them. So at least now we know that PROKKA has been installed and it is working fine. So we have to do the annotation. So let's first get the sample data sets we are going to use. We are going to NCBI to download that genome. We are going to use a bacterial genome. So let's go to the NCBI page. So this is the page where we download the example sequence we will use for this exercise. We are on the NCBI database, and I'm going to leave the link to this page in the description box, so use that to download the sequence. We are going to use the Mycobacterium monstrans aging Y99 sequence as an example. This is a bacterial organism. PROKKA accepts inputs in the FASTA format, so that's the contigs, and so we are going to download the sequence in the FASTA format. And please know that if you have your data in the form of FASTQ, which are the raw reads, then you need to perform genome assembly, maybe de novo assembly, to get the assembly contigs, and you can feed the contigs into PROKKA. And sometimes you can also go ahead to polish the assembly contigs and maybe generate draft sequences from it, and you can use that also in PROKKA. So take note of that. Now we come here to download the sequence. So we use this side here, the send to here. So click on it, and then you have complete record. Check it, make sure it is checked. Come to choose destination here, check the file, and then you have this appearing, the format. We are going to use the FASTA format. So click here and then scroll up to check and find the FASTA and select it. Now we are ready to download. So you click on the create file here, click on it, and then you'll be asked to save the sequence. So I'll save it on my desktop. You can also save it in any appropriate path on your PC. I will name it agy99.fasta. So I have it saved. So this is it, and this is a file. Now FASTA files are just text files, so you can even open it and then just view the sequences here. So but it can make more sense if you use Python to open it and then maybe do some exploration as well. I have a video that shows how to do that, and I will leave that in the description box. So let's minimize this. So now let's go to the terminal and then proceed with the rest of the activity. So this is where we do the annotation with PROKKA. So we are on the terminal. So let's quickly explore the file we downloaded first. So I will say ls. The file is on my desktop. So I will say ls desktop. And this is the file. We have it here. So let's quickly display some lines from that particular file. I will say head desktop / aging y99.fasta. So we have some lines displayed. That's the sequence, some of the sequences, and then we also have the name displayed here. So now our file is ready for use. Let me clear the screen first. Before we run the PROKKA, let's call the PROKKA again. And then we have the option displayed here. So we are going to use a few of these options. We will use the CPUs. We are also going to use Kingdom. We are also going to use the local stack, which is here. And we are also going to use the prefix, which is here. These are the options we are going to use for this exercise. But you can explore these other options. So just get the explanations from this text or from the GitHub page and then try and then modify them to suit your needs. So let me clear the screen first. So now it's time to use PROKKA. So what we do is to first call PROKKA. Then we specify CPUs, which is 4. So the CPUs here will allow PROKKA to use a specified number of CPUs for the genome annotation. So the higher the number of CPUs, the faster PROKKA will run, assuming that those CPUs you have specified exist on your system. So take note of that. For now, let's leave it with 4 so that you can quickly run them. We also specify prefix. We have the prefix here. Let's say aging Y99. So the prefix here allows PROKKA to give a name to all the files that will be generated, in addition to their respective file extensions. So what will happen is that all the files generated as a result of this annotation process will have aging Y99 to their names, in addition to their respective file extensions. Let's also specify kingdom. Let's say bacteria. So PROKKA annotates other organisms apart from bacteria, and it also uses their respective genetic codes. So specifying a kingdom allows PROKKA to use the appropriate genetic code for the annotation. So this time we are using bacteria, and so PROKKA would know that it has to use the genetic code for bacteria. We now specify the local stack here. Let's say aging Y99. So the local stack also gives some form of ID to the annotations that will be generated. And it's very handy, especially if you are dealing with several or different sequences. So specifying those local stacks allows you to easily identify groups of annotations that belongs to a particular file or sample or sequence. So take note of that. So now we have it done. So it's now time to specify the file. We have to give it a full path. So the sequence is in what? Desktop. And it's called agy99.fasta. So we have this file specified. So now we are ready to perform the annotation. But you can review your codes to make sure that you have specified the correct options and you have given the required information. So let's run it. So now annotation has started. So let's wait for it to complete. So the annotation has been finished. And that is why you have this here. it was successful. So let's clear the screen first and continue. So if you do an ls, you will find that a directory has been created. And this is a directory. It was created because of the prefix option we used. This directory contains the files that were generated by PROKKA. So let's ls into that directory. And these are the files that were generated. A description of these files can be found on the GitHub page of PROKKA, so please visit that page. I will leave the link in the description box. So this leads me to the next part of the video, where we explore some of these files to get some information for us. So let's first cd into that directory. Now, we will focus on just this, and then this. So let's begin with this one. The .fa contains predicted proteins, and this one here contains transcripts, or we can also see the predicted genes. So what we have is that this one here was formed from translating the CDS sequences, and so the number here also indicates the number of CDS sequences we have. With the genbank file here, which is the .gbk, it gives detailed information. And so for this particular one, you can use tools such as BioPython, which is a library in Python, to help you read and extract the information from it. I have videos on how you can read genbank files using Python, and I will leave those videos. I will leave the links in the description box. And this one can be read with spreadsheet softwares so that you can quickly explore it, and I will show you how we do it later on. So let's focus on this one here first. So this is a fasta file. This is also a fasta file. So if you are looking for the predicted proteins, this is where you find it. So let's begin by looking at the number of predicted proteins, or let's say the count of CDS. So to do that, we issue this command grep, which is used to find patterns in names or in text files. And then we specify the file, which is agy99.faa. We are using some simple bash commands here to do the exploration. And then we say wc-l. So what we are doing is that we are looking for the occurrence of this symbol here. Fasta files begin with this symbol. This is a multi-fasta file. And so each of those sequences will begin with this symbol. So we counting the number of this also gives us the number of sequences. And that is why we are using this particular command. wc-l will count the lines, because when we do the counting, bash will return everything, all the heads in different lines. So once we count the lines, we know how many sequences are there. So let's run it. We have 5471. Let's do a head command to get the first 5 lines of this file so that we can better understand it. So this is the first 5 lines. These are the predicted proteins. But of course, during the exploration, it's not just about counting the number of sequences. We can look for specific names. Let's just say we want to look at the hypothetical proteins. We can say grep hypothetical. Let's add hypothetical, and we indicate the name. and we run it. This will give us the hits. But then, because there are several lines, one of the things we can do is to just do the count. So if you are to do the count, this is where the wc-l comes in. So we can just issue the same command. Bring our pipe and say wc-l. So now we have what? 2516 hypothetical proteins. We have them here using this here. We can also go into details to find specific proteins. So let's say we are looking for SIGM. We can say grep SIGM. I will use this to demonstrate something here. AGY99.FA. Now, when we issue this is what we get. But these are what? Amino acids. And so it means that there's something wrong with our query here. Bash is case sensitive. And so you need to make sure you have specified the correct name here. So here the correct name is this one. And so we get the hits again. So if you are using the grep, you can also choose to ignore the cases. Then Bash will return any hit, no matter the case it is in, whether it's lowercase or uppercase. So in this instance, if let's say we did this command here, -I, to ignore all cases. Let me issue it correctly here. It's lowercase here. Then we are going to have all the names appearing, regardless of the case. So now we can know what is happening. But you should be careful so that you don't put in wrong terms. So when using the grep, try and then use the appropriate or the correct terms that you are interested in. So these are some exploratory activities we are doing with just the bash script. So that is for the .fa files. Let's go to the .ffn. If you're looking for the number of transcripts, or let's say the predictor genes, number of predictor genes, let me put it that way, then we can also use this one here. So we can do a grep. It is also a fasta file. So I have videos that shows how you can read fasta files using Python. So I will leave those links in the description box. So please try and then watch that as well. So we can say grep and we indicate this one here, and we say wc-l, and we have the counts there, 5523. Let's look at another one here. Let's use a term. Let's just use any term here as well. We have gen. Let's do some playing here. We have some information here. So at least this one, what is returning is not what we expect it to. So again, I'm only using this to demonstrate why you should be careful when using the grep command. So try and be accurate as much as possible. Now, this is for what? The .ffn file. Let's look at the agy.tsv. So this one, you can use a spreadsheet software, but let's just do a quick one here. Let's see head agy99.tsv to get it. So when you view on a spreadsheet, you have the local stuff, the feature type, the length, the gen number, product, and other information here as well, so that you can use that to understand your genomes better. So, and this is just another way to explore this particular file here. But let's open it in a spreadsheet software. So let's go to the desktop and then look at how it appears when we use the spreadsheet software. So this is what tab separated values. Again, you can use Python to also open such files. So let's go to the desktop. So this is my desktop, and this is the directory that was generated. And inside it, you have the files here. We are looking at the .tsv here. So let's open it with any spreadsheet software. I'm using this tool here. And this is what comes up. Let's zoom in here so you can get it better. So using this one, you can also read and then try to get some information as well. So of course here gives you the feature types and their associated or their respective information. So let's even do a sort here, or let's filter, and then when we use the filter, we can just filter based on feature type. So at least this one will also let you know which features were predicted at a glance. So at a glance, we know that CDS, we have this, we have this, we have this. These are the features that were predicted for us by PROKKA. And it is this same information you find in the genbank file. Just that the genbank files give additional information, but these are the features that will be present there, and we have their respective counts there as well. We have for CDS 5471, RNA, tmRNA, tRNA, and all those ones there. So this is also very helpful. So that is it. In addition, I would also like you to check these options in PROKKA. They might be of use to you. The link to this page is in the description box, so please use that to read more about these options. So using PROKKA, you'll be able to perform annotations and then try to understand your genomes better, which is at the end of the day one of the aims of what's doing bioinformatics, doing genomics, doing bacterial genomics, and to help us probably also study and then understand those organisms better. So that will be all for this tutorial, and then I'll see you in the next sessions and give you some additional bioinformatics tutorials. Goodbye, and then have a nice day.